All right. So, has everybody had a chance for all three bags of candy? No? In the back row? What are we missing? Good? Perfect. Well, I'm going to put the extra candy up in front, so at the end of class, if you want some more, go ahead and grab some. So happy Halloween. I threw up a picture of my cat who is wearing her Halloween outfit. Yes, she is adorable. She also kept me awake all night last night, sleeping on my face. So.
Oh my god, for real? I feel like they want to kill me.
She's very cuddly. Very cuddly.
Like on your
Yes, that is true. All right. Any questions content-wise before we begin? Alrighty. So we're going to move into the cell cycle and mitosis today. All right, so hopefully you watched that video. We will do a brief recap of that video here. But we're talking about cell division now. All right, so we saw how DNA replicates. Now we're going to look at how cells replicate. All right, so cells, they're going to rise through the division of pre-existing cells. All right, they don't just come out of thin air. Two types of cell division. All right, there's mitosis and meiosis. All right, today we're mostly going to be focusing on mitosis, and we'll get to meiosis on Monday. All right, but both forms of cell division are going to be accompanied by cytokinesis, where we are actually splitting the cytoplasm apart. We are going to dive into all of this a little bit more, a little deeper here. So meiosis, that's going to lead to the production of gametes. Think of egg and sperm, and that the daughter cells have half the genetic material as the parent cell. All right, so that's a big defining characteristic there. Now mitosis, that's going to lead to the production of other cells, known as somatic cells. And this is where that genetic material is copied and then divided equally. All right, so the daughter cells are identical to the parent cells. All right, so meiosis, we have half the genetic material. Mitosis, we have an identical version to the original. All right, cell division, hugely important here. All right, we're going to replicate that genetic material. We need to be able to pass that genetic material on. And we're going to divide that genetic material evenly to form two cells. So this is an example of a bacterial cell. You can see it's starting to divide here. Cytokinesis is starting to happen. We're splitting the cytoplasm. In single-celled organisms, cell division is important because it can result in asexual reproduction. All right, so splitting of the cells are identical to each other. For multicellular organisms, all right, it allows for growth. So think about an egg that becomes a zygote, and then from there we have these dividing cells that allows for the formation of another human being or another small animal or what have you, all right? So that's very important in reproduction. It also allows for growth as we grow up later on in our life stages. It also allows for repair, all right? So if you've ever broken a bone, this poor person here looks like they have broken, a leg bone it looks like, all right? Because we have the ability for cells to divide, we can repair the broken pieces of this bone. All right, for multicellular organisms, they can also go through asexual reproduction, all right? So they're not going to have an egg and a sperm, all right? They're just going to divide. And in this example, we're looking at a hydra. We have this little budding piece here. It's going to grow and eventually just break off from the parent and be an identical copy genetic-wise. All right, so cell division, super important. All right, we could not live without cell division. It allows for all these different processes to occur. And the basis behind cell division are chromosomes. All right, so chromosomes, they play a key role. And there's a little bit of vocabulary we have to go over when we're talking about chromosomes. All right, so the field of genetics where we can actually look at chromosomes under a microscope is cytogenetics. All right, we can see that here. We're looking at chromosomes under a microscope. When cells get ready to divide, this is when we can see the cells under a light microscope because they're more condensed. All right, so if we look at this image here, there are two cells where we can actually see the individual chromosomes because they have condensed because they're in the middle of dividing. All right, these other cells, yeah, those are chromosomes, but we can't distinguish one from another. All right, so we can see them when they start to divide. And when we look at the chromosomes under a microscope, we can start to look at the karyotype of an animal or of a species. All right, so the karyotype is what we see in this image up here. All right, it's going to reveal the number, the size, and the form of the chromosome in an actively dividing cell. Again, we're saying actively dividing because this is the only time we can see chromosomes because they're condensed enough for us to tell them apart. All right, so if we're looking at a cell that's rapidly dividing, all right, that means that these chromosomes have already been replicated. And these chromosomes are going to be joined together into a pair and called sister chromatids. All right, so sister chromatids we have two sister chromatids on our example in the bottom here. All right, we have one chromosome. They're homologous chromosomes, which we'll go over in the next slide. They're going to divide. We're going to have two copies joined in the center and form a sister chromatid. All right, so that common X shape you see is a sister chromatid. All right, so humans, we have 23 pairs of chromosomes. All right, now 22 of these are autosomes. So an autosome is not a sex chromosome. All right, so we have 22 of them that are not a sex chromosome. We can see them labeled here. They're generally going to be labeled according to size. So you'll probably see the biggest one starting at 1, smallest one ending at 22 in humans. All right, now our 23rd set of chromosomes, those are sex chromosomes. All right, so we have one pair in humans, either an XX if you're a female or an XY if you're a male. All right, and that is down here. So that would be number 23. All right, good on terminology so far. All right, I'm going to throw another term at you here. Ploidy. So when we talk about mitosis and meiosis, we're going to see this quite often. All right, so if cells are diploid, all right, we're going to have pairs of chromosomes. So if we see in this image down here, we have these pairs that are grouped together. All right, so that is diploid. You can think of di equals 2. All right, versus haploid. So haploid is typically going to be a gamete. So we'll see this in like an egg or a sperm, where we only have a member of each pair for the chromosomes. All right, so here we don't have doubles, we just have a singular chromosome. All right, so most species are going to be diploid species. All right, for the most part, most of the cells we're going to look at are going to be diploid unless we're talking about egg or sperm or gametes in any sense. All right, so on diploid species, we have members of pairs of chromosomes that are going to be called homologs. All right, so these are homologs. All right, they're called a homolog because they are similar due to a common ancestry. All right, so they're derived from some chromosome, but they're the same chromosome, but after many generations, variation has been introduced where now they might be slightly different on either side, or whichever chromosome we're talking about. All right, so for example, if we look at the color for the gene for eye color here, we can see that maybe one of the homologs codes for red eyes and one of them codes for purple eyes. All right, so they share a common ancestry, but they have slightly different variations to them. All right, so that brings us to our next term, which is very similar. All right, autosomes. So each homolog, nearly identical in size and genetic composition, is an autosome. All right, so this is a homologous pair. This would be an autosome, and this would be an autosome. All right, good on terminology so far? All right. One other terminology, set of terminology is a sex chromosome. I already mentioned this. All right, this is where we have the X and the Y, XX female, XY male. They're very different from each other in size and composition. All right, so if we look at the XY in the male, for example, very different. So they're not going to be considered homologous. All right, so these sex chromosomes are not homologous. All right. I hope I didn't bore you too much or go too fast with the terminology. But we're going to switch to the cell cycle now, where we can actually put cell division and all that terminology into play. So hopefully you should have watched that video I asked you to watch on Wednesday. I think it was 2 videos. And so this should be more or less review, right? All right, go ahead and review it with your partner. See how much you remember, and then we'll come back together.
One S is then everything like duplicating, and then B is a little more growth, and Emily's gonna make us all go with I wish that I wish I'm doing many at home. What are you doing? Well, you're supposed to at some point.
Yeah, I'm going to at some point. I'll protect your stuff. I'm going home. I can do that?
Yeah, but literally just like filter them by online.
Oh, and I can just go to the crib and just stay.
Yeah, you can stay at the crib. Yeah, show them a list you can get.
You see that terminology around?
It shows like size 16.
15.
Yeah.
All right, so cell cycle. Who can tell me the stages in the cell cycle? All right, who wants to throw out the first stage? G1. All right, what happens in G1?
Puberty of the cell.
All right, interesting way of looking at it. In other words, that means
Growing up.
the cell is growing. Yes, G1, we have cell growth. All right, these are where these cells can prepare to divide by starting to grow. We might also have signaling molecules that can accumulate, that kind of progress the cell through the rest of the cell cycle. All right, so G1 is kind of this preparation stage. We are preparing to grow. All right, next stage. I heard it, S stage. Yes, what happens in the S stage? Chromosome is going to be duplicated, yes. And we can remember S stage means duplication because we are synthesizing new chromosomes, all right? We're creating a pair, and S, synthesize, all right? We can think about it that way if that helps you remember it. All right, so after the S phase, we have another phase. G2 phase, yes. And what happens during this phase? I heard something over there. The cell prepares to divide, yes. Yes, so the cell is preparing to divide by making sure it has synthesized the proteins that it's going to need to bring it through mitosis. All right, so I just kind of gave away the next step. All right, mitosis. And what's happening in mitosis? Yes, we're getting ahead of ourselves here. Many different phases, but the cell is essentially going to divide, right? We've been preparing all this way, and then we finally get to mitosis. The cell is going to divide. At the end of mitosis, sometimes it's lumped together. We have cytokinesis, all right, where the cell actually splits apart. All right, so what is interphase? All three of those together, what three are we talking about? G1S and G2, yes. Together they are known as interphase, yes. One thing I wanted to point out, which I think the video might have briefly mentioned, all right, we also have this G0 phase, all right? So in this G0 phase, these are basically cells that are not yet ready to divide. They're either going to postpone division or never divide again. All right, so they're going to hang out in this G0 phase instead of continuing on. All right, does the cell cycle make sense? Awesome.
Wait, can you go back one?
Go back one, yes. All right, so we have interphase, mitosis, which can also include cytokinesis. All right, and the G0 phase, which sometimes occurs. Now, the cell is going to spend most of its time in a certain part of the cell cycle. Does anybody know what that is? Interphase. Yeah, the cell's going to spend most of its time hanging out in interphase. Any other questions? All right, so what tells a cell to divide? Well, there are a lot of things that tell the cell to divide. We're not going to go too in-depth on most of them here. We could have environmental conditions that will tell the cell to divide. All right, maybe think about a plant. During the summer, we have a lot of plant growth. All right, environmental conditions are telling that plant it's a good time to grow. There might be different signaling molecules. Maybe it's different hormones that might signal growth. We also have internal factors like cell cycle control molecules and checkpoints. All right, so let's talk about the internal factors here. All right, so we have these checkpoint proteins called cyclins and cyclin-dependent kinases, or CDKs. All right, so cyclin is responsible for advancing a cell through the phases of the cell cycle when it binds to these kinases. Right, now it's called cyclin because it kind of cycles through how much we have in the cell. All right, that's where its name is derived from. So the amount of cyclin is going to vary throughout the cell cycle. And then the CDKs are going to be these kinases, or these enzymes that are going to control the cell cycle. Right? They have to bind to the cyclin to be active. All right, so I thought I had another slide after that. All right, so we're going to talk about 3 different checkpoints that involved these proteins, that involve cyclins and these CDKs. All right, so there's 3 checkpoints within the cell cycle. We have the G1 checkpoint, the G2 checkpoint, and the metaphase checkpoint. All right, should be pretty easy to remember where they're located because it's literally in the name here. Now these checkpoints are going to kind of act as these sensors to make sure that the cell is okay to move on to the next step in the cell cycle. All right, if there's any problems, we're going to delay that cell from going through the rest of its cycle. Now it's when we start to lose these checkpoints that we can get mutations, we can get certain types of cancers. All right, so let's actually look at a visual of what these checkpoints look like. All right, so let's start with the G1 checkpoint. All right, it happens in interphase, the G1 step here. This is going to determine if the conditions are favorable for cell division. All right, so we have this CDK. If conditions are favorable, the cell is going to synthesize cyclin. That cyclin is going to bind with the CDK and progress the cell forward onto the next few cell cycles. Now, if it senses any problem, maybe there's mutations in the DNA, all right, this is not going to happen. All right, this is where the cell can basically say, I'm not going to divide because there's something wrong here. All right, so that's the first checkpoint. Now, eventually, this cyclin is going to be released and degraded after it enters into the next phase. All right, the next checkpoint comes at G2. All right, again, we have that CDK protein that's hanging out. And if that protein has determined that, that the cell is okay to move forward, that there's no mutated cells, specifically in G2, that all the DNA has been replicated, all right, this mitotic cyclin is going to bind and tell the cell the same thing. It's okay to move on into the next phase, into mitosis. All right, now we have one last checkpoint during mitosis, specifically in the metaphase checkpoint, all right? This is going to determine if all the chromosomes are properly attached to the spindle apparatus. All right, so if you remember back to high school, there's that spindle apparatus that they're attached to. We'll go over that in just a little bit here. All right, so we have 3 checkpoints. So at this point, before we start talking about mitosis, at the end of interphase, at G2, we are ready for cell division. All right, we have chromosomes that have been replicated. The cell has grown. Proteins have been synthesized. All right, the cell is now ready to enter into mitosis. This is just another image saying the same thing. All right, we've replicated it. They've been condensed down almost to this point, not quite. All right, so interphase, they're still less condensed, and they're ready to go through mitosis. All right, so we've gone through interphase. Let's look at mitosis. I know this is a lot of talking, but I will have a worksheet where you can go through this a little bit more, because I think listening to me is pretty boring when we talk about mitosis. All right, so the cells are going to divide to produce 2 genetically identical cells. All right, so the original is the mother cell. The offspring is the daughter cell. So this involves mitosis, but also cytokinesis. So if we look at the figures here, and I'll give you a chance to look at this a little bit more in depth, let's walk through these phases. All right, so we just exited interphase. All right, we have replicated chromosomes. So the first phase of mitosis is prophase. All right, this is where those sister chromatids are going to condense. All right, this is the point where we can start seeing individual chromosomes under a microscope because they have been condensed. Now the nuclear envelope is going to begin to dissociate into different vesicles in this stage. All right, now in prometaphase, this is where the nuclear envelope has totally gone away. All right, it has dissociated into these small vesicles that are now hanging around the chromosomes. Also in this phase, the mitotic spindle is fully formed, and we're going to attach our sister chromatids to the spindle via a kinetochore. All right, so let's look at the spindle a little bit more in depth. So the spindle is going to organize these chromosomes during cell division. All right, that's its job. It's going to organize them out. Now they're composed of microtubules. These are part of the cytoskeleton inside of a cell. They're basically long cylindrical protein fibers. All right. Now they are going to originate from a centrosome, or the microtubule organizing center. All right, centrosome and these MTOCs, same thing. All right, different terminology. So these centers, they're going to be duplicated during interphase, and when we get into mitosis, they're going to navigate to each end of the pole. All right, so opposite ends of the cell, and this is where the spindle apparatus is going to begin to go inwards. All right, so these spindles are going to form these microtubules again, these long protein fibers, and there's 3 types of them. Right now, I'm not too concerned that you know all these 3 types in detail. but they're going to come up in a lot of diagrams, so I want to make sure you understand what they are. We have those astral microtubules, which are going to position the spindle inside of the cell. All right, make sure they're positioned correctly within the cell. We have the polar microtubules, which are going to separate out the two poles. And then we have the kinetochore microtubules. All right, this is where the chromosomes are going to be attached to the microtubules via this kinetochore. All right, so this kinetochore is a grouping of proteins that are over top of the centromere of the chromosome. All right, and this is where those microtubules will attach. All right, so once that mitotic spindle is formed and attached to those sister chromatids, we have our next phase, which is metaphase. All right, this is where we are lining up all of our sister chromatids along the metaphase plate. All right, so the plate you can think of as a plane of existence. All right, it's not a physical thing, it's just a plane where they're going to line up along. Once they've lined up, this is where that mitotic spindle is going to contract and pull apart our sister chromatids. And this is anaphase. All right, so we're separating out those duplicates. That way each cell has the same amount of genetic material. And then the last phase, telophase. All right, this is where we have the reformation of our nuclear envelope. And this is where we start to see the cell dividing. And the act of actually dividing the cell in half would be cytokinesis, dividing that cytoplasm. All right, so that is going to form two totally new cells. All right, so cytokinesis is different depending on what type of species we're talking about. So if we're talking about animals, this is where we're going to see that cleavage furrow that you commonly see in most diagrams. But if we're talking about plants, they have a different way to break apart, and they actually form this cell plate. Right? So they're actually forming a cell wall in between the two new cells to separate them out. All right, so there is a little bit of distinction between the two. All right, so this is where, enough of me talking, I have an activity for you to do. It's just to make sure that you're understanding everything so far in lecture. I'm going to go ahead and pass it out. Feel free to work with a partner.
Stop. He's so loud. You can only do that.
All right, as you're going through these questions, a couple of them are going to ask you to draw out a cell during a certain phase. All right, to give you an idea of what it might look like for, I think, what did I write, 2 pairs of homologous chromosomes. All right, this image here shows 1 pair of homologous chromosomes after they've been replicated. All right, so that might help guide your drawings a little bit.
The cell cycle is. It's dry. It is dry. That's okay.
It's a boring mustard because it's such
that
dry material.
No, I like the material this time. I
Yeah,
didn't like
this is
actually
this is
good.
where
I think it gets more exciting when
This
we
is
talk
when
about
start to
real.
go.
I just
Yeah. It's not like a bunch of chemicals. It's like actual
That's true.
it's like when I walked in here the other day and we started talking about DNA synthesis and stuff and replication.
Yeah, I know. It's like
I was like
this
stuff,
is stuff
stuff I actually kind of know.
I think every student kind of has that part in Bio 101 where they're like, finally, I get to talk about this.
Yeah.
I remember this.
Yeah. It's just a lot more easy than
I can see that. Yeah.
because it's getting bigger too.
Yes, it
You
is.
can actually see what's going on.
We're beyond molecules. Yeah.
Here, this is our primitive recording.
So one homologous pair would create.
Oh my god.
Oh my goodness.
Yeah.
What's it going?
Yep.
Who's not the two from those together is the just the one. But there's
Individual. Yes.
Yeah.
Okay.
Sister.
So do you want mine to be such a strong girl?
The identical copies.
Do you want mine to be-
Is after replication.
Oh, I hate that.
I know.
This is two.
But like,
Yeah.
just. Oh wait, I don't know what happened. I hate it.
This is so stupid. You get whoa, you get it? There was another one.
I have a question.
Yeah.
What building did you live in your freshman year? This is very important.
Freshman, I lived on Sorry third floor.
I called it.
I wasn't an athlete, so I think that automatically kind of pushes me to this side of the campus.
Oh yeah. It's nice though, because I had like a 30-second walk to the music building right away in the morning.
Yeah, totally.
Did you go through your 4 years?
I did.
And then
I went to grad school in
then I
Ohio.
woke up and it's
Small college.
45.
When they did this in the
I'm committee. I taught at my grad school. I taught at St. Mary's and I was on this committee. On Friday.
That bad.
No,
Like,
it's bad.
it just makes
I have another question. Did you graduate in 2016?
Yes.
like, I don't like
Yeah, something of your time.
Oh my gosh, EPS.
It's been. Yeah.
No.
What's that?
Oh, it's an anonymous commenting.
Our guys are weird.
No. I mean,
It smells like ketchup.
I guess it depends on the individual. I mean, I'm just like, bro. Yeah, like, ego challenge. Yeah, not me.
So right now,
Like, not me.
there are 2 big generations.
Yeah, I mean,
My boss's name is Eric, but spelled the most confusing possible.
How's it spelled?
Who's Derek?
Yeah.
It's spelled like this, and then this
Is
is
it B-E-R-I-K?
no. It's even weirder. I'll put it underneath my two emails. This is how they're spelled. It's so weird.
that's normal.
Yeah, I mean, the extra R there is kind of.
Yeah, usually you spell it D-E-R-E-K.
Yeah.
Especially your white and Ohio. So.
My friend there is back home. I don't know how we said his name. Derek. Derek. Yeah.
Yeah, do you want
Hey, were you at the Q?
On Tuesday.
Yeah, do you drive a red car?
No.
There's a red car from Texas that goes here.
And we should know that. I don't.
Yeah, no, all three checkpoints are with
Wait, how do you know what my
Weren't there though.
So in P2, the cells.
Yeah, I think he does right now.
At this point, it's already been
Because
replicated.
it is like.
So any proteins that might be literally.
No, it's maybe some.
No, literally really have those. Yes. But more specifically, they each are here for individual things. Yeah, I would. So if we look at each one there, basically making sure that there's no
Yeah, I would love to.
no mutated DNA, all the other conditions are right. I prefer to have a heart attack.
I get it.
Oh, no
That's our
mutated
time.
huge excellent. We're making sure that.
Well, yeah. I'll be like, bro, what you want?
Yes, it's been all replicated. You got it. Nice. So making sure that all replication has occurred. If it hasn't occurred, we're gonna get some funky daughter cells. And then once we hit metaphase, the metaphase checkpoint is checking to make sure that all the sister chromatids are attached to the spindle. Because if they're not attached, then we're gonna be separating out in ways that we don't want.
Shelly did her glasses yesterday.
Shelly's awesome.
These people fell sick.
Say one's like a G2 checker, then I would expect you to say something.
Oh, I thought that was.
That's why
I have a question.
Looks like it. Oh, great. Fine.
I have a question related to the class this time.
Yes.
Can we get a unit 3 study guide?
Oh, yes. I was gonna let you know. I'm gonna try to have that up over the weekend.
When's your next
Next test.
or exam?
Yeah, same thing.
Next Friday.
Next Friday. It's not next Friday. I think it's the Friday after
It
that.
says quiz for next Friday.
Yeah, that's why I'm saying.
If I don't make them, I'm not screw that. I'm making them.
I need to upgrade grade in this class. I have B plus. They're not gonna like
Yes,
that.
but I didn't have.
That is nothing to be ashamed
No,
of.
it's not. But I have to hold the 3.75. Otherwise, I lose like a ton of scholarships back home.
That's rough.
So.
Back to like what we back to like what we
I have, I think, like
people that are from
$3 grand. Rich people.
they record you and get it back to you on Monday. Otherwise, they're free for you to keep. You have about 10 more minutes to keep working on and asking me questions.
Wait, Charles.
I did want to mention that next Friday we do have quiz 4, so be prepared.
Always on my time.
Shirley's from Houston. I feel like John is from like the first Texan accents. Like,
Would you put telephase and centigrade in like the same category? Or would you separate them?
I need this. Can you check Corey's thing so I can see it?
Because like you said, he's on here, it has them together.
That's like,
Yeah.
when most people
Generally,
think
it
about
happens
Texas, like with all the consistent
at the
Texas,
same
like,
time, really quickly.
Because one telephase should be like,
but like
like
where
they're
I'm
still
from,
together. So it's
Yeah, I'm telling you,
Well,
we're-
it's correct to an extent.
Like
yeah.
molds.
but at the same time, cytokinesis is starting to happen.
why? They're the best.
There you go.
Check Corey's sheet so I can look at it. She's not looking at it.
Make sure you write it down.
It's like a glorified mohawk.
No, it's not.
Yes,
Well,
it is. It's just a little bit.
Professor Newell, what is your, I know this is biology, but this is an
It's
important
biology.
question.
It's kind of biology.
This calls into question like morals.
Thank you.
What do you think of mullets?
Yeah, like not the
Like
fish.
what's your
Like
yeah,
the
like
Hear me out.
Your boat.
Hear me out.
No,
Let's
no,
give
no.
John.
Keep your full.
no, I wouldn't do that to anyone. I wouldn't. Like if I see someone wearing a mullet, I'm gonna be like, dude, I'm really sorry for you. Like it's really messed up what they're doing to
I don't think
you.
mullets are super attractive, but I could see where they were like in style.
No.
So I would say autosome here. Literally around.
Wow. So you're telling me if Justin Bieber had a mullet, he'd be like that? There's no way.
Yeah, exactly.
Yeah,
so- It's just like.
No, there's no shot. He's already like a perfect human being. When you think about it. You have to be better.
They like replicated themselves.
Is that a hand up and a question?
No.
Sorry, I was just looking at it. I felt like we. Alright.
That's not a mullet.
It's a mullet.
That's not a mullet.
Yeah. They're checking for ears.
They're so greasy and nasty. It
Once you have finished, you are free to go and grab another piece of candy if you'd like.
listen. No, it is. I'll get like fucking sugar.
I get mine really.
Yeah, listen. We all have our things that we're just like, yeah, like we get done. Listen. I'm okay.
You know, if it means that I'm gonna get better ads, I don't want better
Hey,
ads.
listen.
I want so everybody
It's a
is.
plus. And here's the thing. I have a beginner, but I can't track.
Get this. I got all my tracking stuff on, and I got an ad for Roma Ducky ice cream.
Like that's cool.
Exactly.
That's really cool. For real? I'm bored.
Hey, what time are you gonna be back?
6:30. don't know.
Wait, Anna, you left that phone in there?
I don't know.
I really doubt it.
Every time you say that, you're like, oh wait,
No, it wasn't. I checked. I
There's not a
checked.
class in there right now.
No.
So you could just go check.
I got two.
Have a good Halloween.
Yeah, you too.
Get it?
Maybe. Looks like it. Go for it. I'm gonna get trick or treaters at my apartment. Really? Really?
Sorry. Instead of bringing it to the locker room.
Wow. Well, I'm curious. You gonna be here for a while, Kat?
Email?
Yeah.
Did you just send me one?
No, I like you sent me one.
Oh, yeah. After we met, I sent you one. Just so you kind of have an idea where you are.
I like made up my mind.
Okay. Okay.
Yeah. Do you have like the do you like get form?
You have to get on the register.
I was just there and they're like, I don't know if they just didn't have it for me.
Alright. I'm not sure. Since this is my first semester here, I'm not sure how that process works, but I will look into it.
I can always go back and see too.
I thought I was in the registrars. It might be a form you find online. I'll have to do some digging.
Okay.
Yeah.
I'll look online too.
Sounds good. I'll send you an email when I find it.
Okay, thank you so much.
Have a good weekend. You too.
I assume you probably have things planned out a little bit in advance. Do you know what what next Friday's homework load is going to be, or if there is going to be any?
Next Friday's homework load. Let me look at it. Next Friday. Oh gosh, that would be the 5th.
So if there was the quiz, so probably nothing after.
There might be a reading or video I have you watch.
Okay, but nothing too much.
Nothing too much.
Okay.
And if it is
Sorry,
something,
you're fine.
it's going to be one of those, if you did it, you get the points. So,
Okay.
yeah.
All right, I was gonna say because got an email today from Clay for the routine and for Christmas card chapel. He was like starting Friday night, and then he was like 4 hours Saturday morning, 4 hours Saturday evening. So we get like the one weekend, and I'm like, but I have things.
That's pretty rigorous.
So now I'm pretty asking really, hey, quick question. Do you know what's happening next week?
Yes, yes.
Great, thank you.
Are you
Yeah.
gonna be doing a study guide soon for the exam?
I'm hoping to get it out this weekend.
Thank
Yes.
you.
Okay, sounds
Yeah,
good.
have a nice weekend.
Thank you.
Yes.
Adios.
See ya. Still making sense?
Work yourself.
Yeah, I thought so. I thought it was a better way to learn than just talking at you. What made you choose biology? I like biology. I like being outside. I think that's what led me to it. So I'm more like the outdoor environmental biology aspect of things. But it's all pretty cool.
And I'll be trying to think if I should do chemistry or biochem.
Yeah.
Just because I want to go into more medical research type things, I can probably put that might be a better idea. I don't really want to be with companies looking at their waste treatment of chemicals that they produce.
Yes.
But
Toxic waste. Have you looked at J-terms? Are you going to take one?
I'll probably do biomed. Yeah, you have to the freshman. We do biomedical research hopefully.
I was going to say, you should do biomedical research.
Biomolecular research.
Biomolecular, yes.
I emailed her and it sounds like a lot of work, but you have to do like 5 3-page papers, single space, 11-point font, Arial, 0.7-inch margins. I'm like, okay, but
That's a very specific.
one of my friends like do it, so that'll make it hopefully a little bit better.
Yeah, I think just having that research experience, that'll be awesome.
Yeah. Then I feel like, oh, I actually don't have to do this.
Exactly.
Yeah, exactly.
It's only like, what, 3-week commitment, if you don't like it, you know.
Yeah.
3 weeks. J term is how I decided I want to go environmental versus medical. With the J term, like, this is awesome. I want to study this more.
Yeah.
So
You've gone to the field.
I didn't. It just January kind of sucks for that.
Yeah.
But we got to talk about national parks and like environmental impacts and all that fun stuff.